I had the pleasure to share a summary of what my research is about here at UTS. We focused on the development, the bioengineering of heart tissues that may be used for different applications, primarily for overcoming challenges in transplantation of heart tissues for heart failure patients. We focus on understanding what is the underlying problem, meaning what is the cause that leads to a failing heart in a patient. And this leads back to the fact that the lack of proper vasculature is not functioning in a patient that is suffering from a heart attack. Therefore, we try to identify a way to promote new blood vessel formation by using bioengineering tools and stem cell technologies. Now, my research has been focused on, in the past years, on 3D bioprinting of tissues, primarily trying to understand how to bring cells together in what we call biological inks or bioinks. The bioinks that we formulated are composed of cells that replicate what similar cells do in our body. And we took advantage of the use of stem cell-derived cardiac cells, so to mimic a heart tissue in a petri dish. Now, as I mentioned to you, the vasculature, the new blood vessel formation is critical for a proper heart tissue, healthy heart tissue in the body. And that leads also to the fact that there is the occurrence of pathologies. One is a myocardial infarction, but many others that are consequences of the malfunctioning blood vessels in the heart. So we try to understand what was the optimal way by looking at human heart biopsies here in Australia, back in Harvard Medical School, where I was visiting in 2016. And primarily we focused on the understanding of different ways to basically try to understand how it is possible to bring together the cells in the embryo. So bringing together expertise from developmental biology and also by observations from the human heart biopsies, we formulated our cardiac bioinks that replicate the mini vasculature found in the human heart. I'm happy to share more details about what we've done, but I can tell you that the research that I presented here at TERMIS recently is based on the accumulation of almost 15 years of research based in the United States and more recently in Australia since 2013. And I would like to highlight the fact that it's a multidisciplinary research that has been conducted thanks to the support of different experts from different areas. So for instance, my background is in pharmacology and cardiovascular development. I was able to gain knowledge and expertise in bioengineering during my time, my PhD in the United States. At the same time, the research team has been enriched by individuals and researchers that have different background. For instance, one of my PhD students, Chris Roche, is a cardiac surgeon training himself. So together with him, we look at how to implement the transplantation of the heart tissues that we generate in the lab. And then we have other experts in biomaterials that have been tested in order to promote optimal cellular viability and function before and after transplantation. And also experts that have different scenario background in, for instance, in pharmacology. We've utilized bioprinted heart tissues to mimic, for instance, what would be the behavior of the human heart following the exposure to drugs, for instance, or even more complex scenarios where we try to mimic a heart attack in a petri dish. And it's been the body of the research that has been presented at TERMIS a few weeks ago.